Good afternoon, everyone. First, I want to thank the organizers for inviting me to give this presentation. It's a real honor to speak with you today. My name is Michael Whitfield, and I'm Chair and Professor of Biomedical Data Science and Director of the Center for Quantitative Biology at the Geisel School of Medicine at Dartmouth. My laboratory has worked on scleroderma for the last 20 years, and I'm very excited to tell you about the work we've been doing today. The title of my talk is Toward a Better Understanding and More Effective Treatments for Scleroderma, a journey from the laboratory to the bedside. So this is not an audience to whom I really need to explain what scleroderma is. Here we are at the end of the conference where you see many different presentations on the disease and heard lots of really great science and clinical information. But I want to point out one of the major things that my lab has tried to work on, which we came into this knowing that scleroderma was this progressive chronic multi-system disorder. At the time we started working on it 20 years ago, there was very little understanding of what the root causes were or mechanisms that led to the dermal fibrosis of skin that we see here. We're well aware that some patients get internal organ involvement that can be very bad, and other patients do not. There's all the vascular abnormalities and vascular problems that many patients experience, as well as autoantibody formation. And of course, there is a significant mortality, although our treatments have done have done a very good job of making that significantly better. So really what my work has tried to do is take scleroderma and try to understand how the disease is caused, and also to understand the heterogeneous and complex presentation. So we know that different patients experience many different end organ complications, and we've shown some of those here with interstitial lung disease, pulmonary arterial hypertension, renal crisis, gastrointestinal complications, as well as skin fibrosis. And we know patients experience this differently, with some patients getting severe interstitial lung disease and some patients not experiencing that. And we also know that the male to female ratio of the disease is strongly skewed towards women, with a ratio ranging from 4:1 to 7:1, depending on the studies that have been examined. There are only 2 FDA-approved treatments for scleroderma, mainly for scleroderma ILD, and those have only been approved in the last couple of years. So we really need to better understand what's causing the disease, but also how we can treat patients with the disease. So what I really want to talk about today is the idea of precision medicine, and which patients should get which therapy and at what time. And to try to create a framework by which we can do precision medicine in scleroderma, we're going to talk about genomics and genomic data, and you may have heard of the Genome Project, and how that can be used to help patients with the disease. So what is precision medicine? Precision medicine is getting the right drug to the right patient at the right time in their disease. And really the idea is, can we tailor treatments to a subset of individuals with scleroderma instead of having a one drug fits all approach? Imagine if we could do a test that would actually say, for this individual, they should get this particular drug and it's most likely to treat their disease, rather than a trial and error approach where you have to try it, take a couple of months, see if it's impacting your disease before you change to another drug. And what we're going to very specifically do is ask, can we use genetic or genomic information that could be measured from individual patient skin biopsies or blood draws to better understand scleroderma and determine who will respond to a specific set of treatments. So I mentioned the genome. So I want to step back and say, what is a genome, right? So you've heard this term before. You've probably read about it in popular media and heard about it in maybe movies, but let's talk about what it is. So we can go to the Oxford English Dictionary and actually look up what a genome is, and I've pulled that definition here. And what you can see is that first, this is not a new term, even though we talk about the genome being fairly new to science, but it's really something that we've known for a long time, and it's all the chromosomes that are in your cells, right? So these are our DNA that's in every cell, almost every cell in your body, and it's all the genes within that DNA, right? So this is a lot of information. The very first use of the word was in 1920. So it's gene plus chromosome, so genome. And you can see Oxford English Dictionary gives citations for the word in 1930, 32, 65, and 70, and to modern times. And one of the things that we can do is you can see how far our understanding has come, as I'll illustrate in a couple of minutes, is that in 1970, where we had not been able to sequence the human genome at all, it was stated that the human genome consists of perhaps as many as 10 million genes. So we know that's a number that's really far off at this point, and we only have about 22,500 genes. So the genome, that's in each of your cells, is constructed from DNA bases, which are the building blocks of the genome. And there's only 4 letters in your genome. It's a very simple alphabet, only A, T, G, and C. And we know that these form base pairs in what you may have heard as a double helix. So A is always paired with T, G is always paired with C. So to give you some idea of the amount of information that's in your genome, the total genome length, and this is just for so one copy of each of your chromosomes, is 3.1 billion base pairs, right? So 3.1 billion is a huge number. And to give you an idea of what 3.1 billion is in terms of a number, I have a couple of examples. So for example, if you were to read the genome sequence, base by base, without stopping, it would take you 9.5 years to read it from end to end. If you were to print the genome in a phone book, in say the Manhattan phone book, it would take up 200 copies or more of the Manhattan phone book. So you'd have all of those sitting in a room, and you have to sort through all that information. And if you were to put it onto a hard drive on your computer, you're talking about 3 gigabytes. So that's a lot of information, but it can also fit on something as simple as your cell phone at this point, which contains quite a lot of information. Now, one of the key things is that although we have this genome in all of our cells, and it's 3.1 billion base pairs, if you were to look to the person next to you, or any other random person on the planet, you can find that each unrelated person varies at 4 million of these base pairs. So, but that sounds like a lot, but really what it means is that only 0.1% of our DNA is different from between 2 individuals. So you're really not very different from any other person on Earth, but we can find things in there that predispose you to disease and may make one person's scleroderma different from another. Now, we're not going to talk about the genetics, which others have done really fantastic work on in scleroderma, where this is changes in your DNA that may provide a risk for the disease. But we're going to talk about the genes that are encoded in the DNA. And your genome, I mentioned in the prior slide that the estimate in 1970 was that your genome contained 10 million genes. And what we actually know from the Human Genome Project, which has sequenced the entire genome, is that it really contains 22,500, roughly, give or take a few, genes that are distributed throughout this sequence. So that's many fewer than we thought. But we can think of the genome as a map and the sequence as a map. And it's analogous to the map of the world. So here's a map of the world in 2003. And what you can see if you look at this map is there are continents, there are oceans, there's the poles, and there's all sorts of land masses and structures that exist across the Earth, which we can document very clearly. And you can go to the map and find where things are. It's a lot of information, but it's searchable. And you can actually, if you want to know, if you did not know where a city was in the United States, you can go to the map and look it up. Now, just like the genome, we know that our continents and land masses and bodies of water are changing at a very slow time scale. So geologic time. We also know the genome is changing at very slow time scales when it comes to the DNA level. So we know that mutations occur, and that's clear in things like cancer, but this is a slow process in many cases, at least the normal process of genetic variation. But we can also take and now look at a genetic map, where here I'm showing you all of at least a representation of the human chromosomes that would exist as part of the genome. So I'm showing you just a subset of these here, and what I've highlighted are a subset of genes, or what the figure has highlighted, are a subset of genes across these chromosomes. This is not all the genes that exist on the chromosomes. This is just a very small subset, and each of these has names. And these names, will not mean anything to most of you. Some of them don't mean anything to biologists. We have to look them up because there's a very large number of them. But we can measure the expression, or at least if these genes are turned on or turned off, using a couple of technologies that came out of the Human Genome Project and innovations at other institutions. So one of those innovations, which is where I got involved in this work, was something called a DNA microarray, where you could take the DNA that corresponded to each of those genes on different chromosomes, and you could spot them all onto a glass slide. And you could do this for every single gene in the human genome, right? And then you could ask, are these genes turned on, or are they turned off in a specific sample? So say a skin biopsy from a patient with scleroderma versus healthy control skin. And these are all fluorescently labeled, so they light up like a Christmas tree. And when you actually do the experiment, what you can begin to see is, if you do this on blood, and this is a blood sample, is every single one of these little spots is now the level that a gene is turned on or turned off in your genome. And in this case, what you're seeing is, if it's red, that gene is on in the blood sample. And if it's green, it's effectively off in the blood sample. And you can actually collect all this data using a computer, put it all into a giant spreadsheet, and we can actually zoom in. And you can begin to actually use this information to understand what is occurring within a sample. So as I mentioned, if it's red, it's high, the gene is turned on. If it's green, the gene is turned off. But what this allows us to generate then is a different type of map. So this is a map of the Earth now, similar to what I showed you before, but it's the Earth as viewed from space. And what you can see is that there are, within the continents, there are very clearly distributions of light which are not even. So first, you see that the eastern seaboard is very dense with lights and populations. You can see the West Coast has populations that are very dense, where there's places that are people. And across various parts of the world, you can pinpoint these places. And then you can see places where there's not a lot of of lights. And if you were to do a time lapse and look across this over night and day, across the course of a day or a week, you would see lights come on and turn off and back and forth as people go to bed and wake up. And this is similar to the type of map you get when you look at genes across the genome. What you find is there's portions of the genome where there's lots of genes and portions of the genome where there's very few genes. And you also find that those genes are sometimes all turned on, and sometimes a whole bunch of them are turned off, and you can find patterns to them. And you'll find that all the genes that turn on at the same time are involved in possibly the same process. So for example, it may be they're involved in metabolizing nutrients which you've brought into your cells by eating. And they may all turn on at the same time for certain to metabolize those specific products. Similarly, in something like scleroderma, we would expect that all those genes that make your tissue fibrotic and hard may be turned on within the scleroderma diseased organs rather than being turned off as we would hope they would be in healthy tissues. So we can actually ask these questions. So one of the things that we can do is we can take all of that data and we can put it into a giant spreadsheet. But because your eyes are not very good at reading a giant table of numbers, we've converted these into a very nice color-coded diagram. And these are called heat maps. And the simple way to look at this is that every column is a patient sample. Every row is a different gene. And then rather than putting a number here, what we've done is color coded it. Is it down or up? And the colors are a little different than what I showed you originally. But now it is, if it is blue, it is down. And if it is yellow, it is up. So this is a much higher throughput channel for your brain to read information than the number reading channel. The color reading channel is significantly better. But there's also lots of genes, so we can actually name them. And then we can actually say what they do. So we can say maybe that gene is involved in fibrosis, maybe it's part of a signaling molecule, and then it also may be someplace in the cell like the nucleus. So we can find out lots of information about what these different genes do. So what we'd like to do though, is take all that information and ask, can we understand a human disease? In this case, the disease we want to understand is scleroderma. But I want to give you an example from cancer. So when I started doing this work in the early 2000s, my colleagues who were sitting right beside me in David Botstein's lab, where I did my postdoctoral work at Stanford, were working on various tumor types. And one of those tumor types was breast cancer. And one of the early observations was that you could take a tumor, you could grind it up, and you could isolate the material that comes from a gene, RNA. And then you could actually ask if the genes are on or off. And then we could go through and take a large set of tumors, put them together, and use a computer algorithm that puts things that are similar together on the branch of these trees. And then ask, do we see patterns? So one example I'm showing you here is that if you were to look across a large set of 115 breast tumors, you find 1, 2, 3, 4 different subtypes of breast tumors. And what was really important is they are determined by which genes are on and which genes are off. And they also correspond to biology that we are well aware occurs in breast cancer, such as amplification of the ERB2 HER2 new gene or estrogen receptor positive tumors. More importantly, we can also go in and look at these different groups and show that these patients have different outcomes, either if you look at time to metastasis or overall survival. So you can see that patients who were in this luminal group actually had better overall survival and better outcomes than those who were in the basal or ERB2 subtypes. So this has become very important, and there's now diagnostic tests which exist for breast cancer, which can be used clinically. And we wanted to ask if we can do the same thing in scleroderma. So to do that, I want to step back and just say, how did we put those tumors together onto the branch of the tree? What was the algorithm by which we did that with? And what's the computer doing? And so to illustrate that, I wanted to use an image. So this was a set of slides which Michael Eisen, who's at Berkeley, who popularized this technique for using these types of data, has used for many years. And you can take the image and reduce the dimensionality by making it black and white. So we can remove the color information. And then we can take that image and we take every row of the image and we randomize it. We essentially move every row in the image somewhere else, right? So, and then we can actually reorder those rows and say, how do we get this back? So we take this randomized image and we apply an algorithm that tries to take every row and place it near the row that is most similar to it, right? So it's a similarity. And if we do that, and we place those similar rows together, we begin to get the image back. And you can see there's a tree, you can see there's a shoulder, but it's not quite there. So you can further actually optimize this by, you can take nodes and you can flip them around such that everything is ordered a little more optimally. And when you do that, you get something that's really close to the original image. So you're essentially being able to, you can tell what the image is, and you can make out pieces, but you also do see that there's a few errors here and there. So it's not perfect. But this is what we do with gene expression data and how we begin to analyze this to understand if there's groupings within the patients. So here's just a example with real world data where you can, if you first look at all the genes and how they're expressed within a set of samples, you can apply this algorithm and organize them by putting all the genes together that are doing the same thing. And then you can visually begin to pick out patterns. So clearly this group of genes is expressed together in the middle samples. These are here in the samples on the right-hand side. So we can apply that to scleroderma. So here was one of our first attempts when I first started working in scleroderma to understand the disease. And in this case, we took scleroderma skin biopsies and we analyzed them using all the genes in the genome. And then we asked the computer to simply say, is scleroderma skin different from healthy control skin? And the short answer is yes. There's a very clear separation of what's healthy control and what's a scleroderma skin sample. But you can also see that in this case, we took a forearm biopsy. So these were clinically affected samples. These patients had fibrosis on their forearm. And we also took a back sample, and this was a lower back sample. And this was clinically unaffected. But what was really surprising was that unaffected back skin, so it's supposed to be clinically unaffected, always grouped side by side with the affected sample in this small number of patients that we first analyzed. And this was true for each of these scleroderma patients that we looked at, including patient 4. They were always side by side. And that began to tell us the disease is truly systemic, that we are actually seeing changes within these patient samples, even when it doesn't look clinically affected. So this says this is really a systemic disease. And then if I show you those gene expression patterns, and here's a subset of genes where, again, yellow are things that are on and blue are things that are off. And there's a bunch of gene names here, and I don't want you to get bogged down in what these are. You can just see that the scleroderma tissue is quite different from the healthy control tissue. And that's not surprising if you have fibrosis in your skin, that you're going to have it quite different from the healthy control skin. But what we really wanted to ask from that initial study when I started my lab here at Dartmouth was, can we take skin biopsies from a large set of scleroderma patients? Can we then analyze them using a either gene expression DNA microarray, or now we use a different technique called RNA sequencing that I won't go into in great detail, and then collect the expression of all those genes in the genome and use the computer to say, do we have sets of patients that look similar to one another, right? So is it one disease, or might we have multiple diseases within what we call scleroderma, and specifically systemic sclerosis, or even in this case, diffuse systemic sclerosis? So what we find is that if we look at patients with systemic sclerosis, and this is diffuse systemic sclerosis and limited systemic sclerosis, we find that we can divide them into 4 different groups based on the genes that are turned on or turned off. And these are a lot like fingerprints, right? So every fingerprint is unique, but unlike a true fingerprint, this is not the perfect analogy, which every fingerprint is unique, there's only 4 fingerprints that we can really find. And we can maybe find a few more. We can maybe subdivide these a little further. But the ones we find reproducible is that there's 4 fingerprints based on these genes that are expressed. We call these the intrinsic subsets because they seem to be intrinsic to each patient. And so if I were to measure the forearm and back biopsy of a patient, they're always, or not always, but very normally the same subset, more often than not. And these subsets can fall into these 4 categories. One is called the fibroproliferative subset. And we see cells that are dividing, that they're in the skin. There is an inflammatory subset, which we'll talk about in a little more detail. And these are patients that have a lot of inflammation. There's very, very active disease. And then we actually have a normal light group, which is patients who have disease. They have fibrosis of their skin, but molecularly, they look like healthy controls. And we think that may be a place where someone's disease has really gone to quiescence and is no longer active. So this inflammatory subset, we think is quite important in terms of driving disease, particularly skin disease. So one important point is we've identified these in multiple groups of scleroderma patients. This is not now a one-off observation, but we've actually seen this in every cohort that we've looked at, and we've looked at more than 10 different cohorts of patients. So these seem to be highly reproducible. Also, we found these in multiple tissues. So we've looked in different tissues. So we've looked in the gastrointestinal tract, we've looked in the skin, we've looked in lung, we've looked in blood. And you can find them in each of these different organs and tissues that we look at. So they're reproducible and they exist across different organs. So what do they mean? Right? So these groups we think are part of the process of the disease. And this is one hypothesis. There's actually several different hypotheses about how they are related to one another. But I'll show you just one, which I think is the simplest hypothesis right now for this purpose, which is that we know scleroderma is very likely the result of predisposition in someone's genetics and their changes in their genome, combined with some environmental trigger or some trigger within the body that results in maybe starting down the road of these different subsets. Inflammation is clearly a process that patients experience, and we can see that in our molecular data. This fibroproliferative group, and this may represent damage that's occurring. And then we clearly see in this fibroproliferative group continued fibrosis, but maybe there's additional repair processes that are attempting to occur. And then this normal like we think may be where these 2 groups eventually get to when a patient goes into a spontaneous remission, that this is a point where their disease is less active than it had been before. So one of the things that my lab is trying to do is to subclassify scleroderma patients in both clinical trials and we hope within even routine clinical care using these gene expression fingerprints or intrinsic subsets. And so the goal is to quantitatively subset patients and determine if there's a different molecular response to therapy. And with the hope that we could actually say, if you fall into the inflammatory subset of scleroderma, if we were to take a skin biopsy or peripheral blood sample and analyze it, then you should get this particular therapy. And these are so early, these are still studies that are ongoing, but we have some very positive data that I'd like to share with you today. We have data from the published literature that suggest these different groups will respond to therapy. These gene expression fingerprints can identify these patients, and we know that there's some changes in how they respond. So for example, in a trial with a colleague, Monique Hinchcliffe at Yale, formerly at Northwestern University, she's currently at Yale University, we've been able to show that the patients who are most likely to improve on mycophenolate mofetil are those patients who are in the inflammatory subset. And we find that the fibroproliferative patients are less likely to improve. Also, I'll show you examples from abatacept, where we've been able to show that inflammatory patients also are those that improve on therapy, and we don't see a major impact on those in the fibroproliferative subset. But stem cell transplant, which we have been lucky enough to be able to study patients from the SCOT trial, the stem cell transplant or cyclophosphamide treatment trial. We find that patients in the inflammatory and fibroproliferative improved, although the fibroproliferative patients showed the most significant improvements, where normal-like patients did not seem to show a significant benefit from stem cell transplant. So our hypothesis and our working model is that this substratification in drug selection may be helpful for identifying those patients who would be improve on a therapy. So this just subset is both important and instructive, at least at baseline. And if we actually combine this with genomic analyses before and after a given process or a given treatment, then we can begin to understand if a drug is working and if it's targeting what it's supposed to target, right? If the pathways that it targets go off. And so the other final point that I'll make before moving on to a couple of examples of other data is the question of, we've identified these groups in scleroderma, and do they exist in other autoimmune diseases? And there was a very nice paper from a group in Spain, which looked at 7 different autoimmune diseases and looked at peripheral blood. And the groups they found were not precisely the same as the groups we found, although there are probably overlaps that we need to look at the scientific and research level. But what it fundamentally began to show is if you took patients with rheumatoid arthritis, lupus, scleroderma, Sjogren's syndrome, and other related autoimmune diseases and did similar experiments, you could also find groups there, suggesting that these groups may be a feature of autoimmune diseases. And they had some, such as the normal-like group, which they also thought were very similar to maybe a quiescent state, or when patient's disease is not active. So suggesting this might be a broad paradigm that exists across autoimmune diseases. So we can actually identify these subsets using a machine learning classifier. So I won't go in details of machine learning, but essentially we trained a computer algorithm to take these data and identify which subset they're in without any human intervention. So we're able to take a biopsy, we measure all the genes, we run it through this computer algorithm, and it tells us what subset the patient is in. We trained and validated this on hundreds of skin biopsies from large sets of patients and controls. It has a very high accuracy relative to the initial training set. So that means it's 85% accuracy, which is pretty good. We have it so we can work on multiple types of data, depending on how you generate it, because some people in different labs generate data in different ways. And importantly, we don't have to wait to collect a whole bunch of samples to do this on, which is what we typically had to do in the past. But we can instead take one sample, from a patient, put it in this algorithm, and get the result back, and then be able to state what the subset assignment is. Now, what I want to give you are two examples of clinical trials in scleroderma where we've been able to do that. One is, the first example I'll show you is for the ASSET clinical trial and the intrinsic subsets. And this was a trial to understand if the drug abatacept, which prevents T cell activation, so it reduces the immune response, would be effective to treat patients with diffuse cutaneous systemic sclerosis. There were biopsies collected before the trial started, 3 months into the study, and 6 months into the study, but clinical data was collected for 1 year. And then we measured all of the genes in every sample. So each patient had a date of the biopsy, it was sent to my lab. We did all these molecular characterization and assigned to the subsets. And then we send those subsets back to our colleagues running the clinical trial, Dr. Dinesh Khanna and his team in this case, to analyze the clinical data and ask if there was a benefit for a specific group of patients. So in our hypothesis, our overall hypothesis was that patients who were in this inflammatory subset would be the most likely to show an improvement in their skin disease or modified Rodnan skin score. And so we looked at the change in their skin disease over 12 months, and we found that if we compared abatacept versus those who were treated with a placebo, so not getting any drug in this case, the inflammatory patients really showed a lot of benefit. And the normal-like patients showed some benefit. This is shown graphically on the right, where those treated with abatacept are a solid line. You can see their skin's disease gets better, whereas those who were treated with just a placebo, so not the drug, actually had an increase in their skin score. And so importantly, we did not see any change or difference in the skin disease for patients in the fibroproliferative subset. So suggesting that abatacept is really most effective on patients in the inflammatory subset. This was a phase 2 trial, so this was a study that we completed about a couple of years ago. So this is not currently available for treatment, but is still something that is being studied. So the second example that I want to give you is the SCOT trial, which is scleroderma, cyclophosphamide, or transplantation. Keith Sullivan is the physician who led this trial, and we were very excited to be a part of it. And in this particular study, we found that patients with very severe scleroderma, or this particular study enrolled patients with severe scleroderma, and then they received either 12 doses of cyclophosphamide or a myoablative hematopoietic stem cell transplant. So essentially had their immune system completely reset. And the participants were followed for 5 years, and then they were evaluated for what we call event-free survival. So we essentially said, how well did they do over 5 years? And events were defined as respiratory, so lung, cardiac, so heart or kidney disease, or death. And so what you found is that patients who received the transplant had significantly better overall outcomes and did really well. So what we get asked was, if we were to look at gene expression, so from these patients, in this case, we use peripheral blood, we really wanted to know, did they do better if they were in one group versus another? So we analyzed most of the patients in the study. We analyzed them over time, and we analyzed intrinsic subsets and clinical outcomes. So we just put them into these subsets. So here is just the number of patients in each of the subsets by treatment arm. You see it's small numbers. There's no significant difference between the arms. But one of the really interesting results was if you look at the outcomes for these patients, you can see that the normal light group, there was not a significant difference between transplant versus cyclophosphamide. Interestingly, those patients who were in the inflammatory subset did appear to get a benefit from the transplant, although the difference did not reach significance. And that's possibly because of small numbers or because they got some benefit from cyclophosphamide. We don't know the answer to that. But importantly, that fibroproliferative subset, which is the group which did not respond to mycophenolate mofetil or abatacept, showed a really significant benefit from stem cell transplant, and that was highly statistically significant, suggesting those patients may really benefit from this particular treatment. So our conclusions for this particular study were that participants assigned to the fibroproliferative subset who had tended not to improve on these immunosuppressant therapies were the most likely to benefit from hematopoietic stem cell transplant. Normal-like patients did not seem to show a significant benefit from this treatment. And so, but in the inflammatory patients seemed to show some benefit, but did not reach statistical significance. And I mentioned the possible reasons for that on the last slide. So fundamentally, what we're trying to develop in my lab is this whole idea of personalized medicine, or what I mentioned before as precision medicine for scleroderma. And this would be a paradigm by which we could subtype patients routinely as part of clinical care, identify the correct treatment for that patient based on their subgroup, understand potentially molecular and clinical response, and this is particularly relevant in the case of clinical trials, and then further refine this by asking if patients improve while on treatment. And what does this mean for patients? So where we're using this right now is in clinical trials with colleagues here in the field. And our goal is really to better understand why a trial succeeds, why a trial might fail, and really asking the question of who got better and why. This whole idea of personalized or precision medicine, and can we get the right drug to the right patient at the right time, is something that we're very excited about, and is still something that we're building and developing. And for therapeutic intervention, all these data provide a huge, rich resource by which we can actually identify potentially new treatments for the disease. So we're extremely excited about that possibility, and could we identify novel ways to treat the scleroderma. And finally, I'll just end with a couple of slides that, of how do we begin to, if we find novel treatments for the disease, how do we test them? And one of the things I want to highlight that I'm very excited about is a collaboration that actually came out of the New England Scleroderma Foundation Patient Conference, where I attended the conference, as did a colleague of mine, Jonathan Garlick at Tufts, who I'd never met before. And we both were invited to give a talk at the Northeast Regional Patient Conference. And there we actually met over lunch, very randomly. He is a tissue engineer that builds 3D skin-like tissues in vitro. And he had a graduate student, Mingji Huang, who was starting to think about doing this in scleroderma. And at that meeting, we formed this partnership which has now been going on for several years, where Ming-Ji came to work in my lab, and Jonathan and I have collaborated for several years now to ask, can we recapitulate and reconstruct scleroderma skin in a dish, in a Petri dish? And there's several different types of ways to do this. Individual names here are not important, but what I want you to understand is that we can take a biopsy, take the cells from that biopsy, and try to put them back together in a dish where we can ask a lot of questions about the disease. Here's just one way we've started to do that: is you can you take these cells, which are the building blocks of your tissue, called the fibroblasts. They're what gets really active in scleroderma and produce a lot of the fibrotic skin. And you can put them into a culture dish, and we can grow them, and then we can add the keratinocytes, which are the very top layer of your skin, and the part that's exposed to the surface of the air, and form that top layer. And then we can grow these, and what they begin to look like very much is what you'd expect to see if we looked at human skin. And then if we seed these with cells from a healthy individual, we get a very thin tissue. If we seed these with cells from a person with scleroderma, we get much thicker tissues and stiffer tissues, and we can actually begin to quantify that. So if we look at, this is just looking at thickness. So if we look at those tissues constructed with cells from patients with scleroderma, they're thicker. If we look at how stiff or how fibrotic it is, they're stiffer. And we can also look at molecules we know are important in scleroderma. We can see they're higher in the scleroderma tissues. So this is potentially a tool which we're building out now and trying to add other cell types with the support of a number of different funding agencies that we're really excited about. But what we can also begin to get to in terms of the future of this work is we can begin to ask questions around single cells. We can do this both in tissues from patients as well as these in vitro models. This is these cell culture models. And one of the things is that we have 37 trillion cells in the human body, right? And we don't know what all of them do. There's some of them we have not even identified. And they vary based on whether you're looking at skin or lung. Some of them are the same, some of them are different. And they're things that do very specialized functions. And in terms of what I talked about earlier, was, we were looking at a milkshake, right? We were looking at all these cells ground up together in a single sample. But, what we want to look at is really the fruit salad. So we can pick it all apart, take one cell out and ask what it does. And this gets into huge questions of big data that we're working with here at Dartmouth and other institutions across the country to really begin to understand these data. And just as one example, here's just an example of single cell data from scleroderma, these skin-like tissues we grow. And now in this case, every little dot is a cell, right? So it's not a patient, it's a cell. And you can see that we have some cells that are all together here because they're very similar to one another. These are those keratinocytes on the surface of your skin. Down here are fibroblasts. These are cells that are in your tissues, which are really producing the extracellular matrix that makes the tissue hard. And then here are a few immune cells we put in, macrophages, which we think are key drivers of disease. I don't want to spend a lot of time on this, but this is really where a lot of very important work is occurring in scleroderma, both in my lab as well as the labs of many colleagues around the country in this disease. And there's a lot of very important work going on here from a lot of different groups and different individuals. So I think this is one thing I want to highlight as a really important future direction for scleroderma. So with that, I'm gonna wrap up, and I just want to make a couple of final points and really just state what I think is on the horizon for scleroderma research. And these are my opinions; they're not comprehensive in any way. There's lots of things that need to be done, and this is just a subset of things I'm thinking about doing for my group. And one of the things that we're doing, for example, is we're really doing a lot of work in artificial intelligence. And how do you find new ways to subset patients using either blood, which is much easier to collect than skin biopsies, or even just using routinely collected histological images, right? So if you have a biopsy taken, that goes to the pathology department, and they get images of the skin that we can look at very easily, and use ways to try to stratify patients in that manner. And there's some successes that are occurring. As I just mentioned in the last slide, single cells and organs affected by the disease are really an important focus of a lot of different groups, and really trying to understand what cells are driving the disease and how we can target them with new treatments. And finally, we need to develop new approaches to identify novel treatments, right? How do we really target the things that are driving the disease that have gone wrong in the body? And there's a lot of and that can be either using these skin-like tissues I just showed you, but also ways that collects all the data in the public domain that all the different groups working scleroderma in the world have generated, and begin to analyze those data in aggregate. And there's a lot of different groups, including my own, who are doing work in that area. And I think that's very exciting. So I think all of this leads to potentially, really exciting things in the horizon for scleroderma. And I think there's a lot of optimism for the coming years. So with that, I want to thank my lab for the fantastic work they do. They are the ones in the trenches who are really doing this work on a day-to-day basis. Second, and these names are listed here as well as our collaborators, I want to very importantly thank our funding agencies. My lab has been supported by the Scleroderma Research Foundation since I began working on this disease, which has really been a lot of years, and I really appreciate their support. But of course, we can't do this without a broad range of support, and my lab has been funded by the Scleroderma Foundation as well as the Falk Medical Research Trust, and a number of NIH and government institutes. So we've received funding from NIAMS, which has been extremely important to carry forth the work that we do, as well as the Department of Defense. We've had several grants from the Department of Defense, and we're just starting a new one, which we're very excited about, very focused on this precision medicine question. But all these funding agencies together are critical for keeping the research going. Of course, I thank the colleagues with whom we work on a regular basis, and this is a subset of the individuals who contributed to work I showed you today. And I really want to thank them for all their work. And then most importantly, is a thank you to the patients who have generously given of their time, donated samples, and participated in these studies. We could not do it without the patients. So thank you for everything you do to make research in this disease possible. We really appreciate all your efforts. So thank you very much for listening and watching, and I'm happy to take questions. Thank you.